Today's video is on skeletal muscles. So first we're going to have a quick overview of the three types of muscles that are found in our body. So the first is cardiac muscle. So this is found in our heart, and this is under unconscious control because this is contracting and relaxing without us having to consciously control it. We have smooth muscle, so this is found in the walls of our blood vessels and in our gut, and this again is under unconscious control because we don't have to think about wanting to contract the muscles in our gut or our blood vessels. And then we have skeletal muscle, which is the subject of today's video. So this is attached to our bone and it's under conscious control. So this is what allows us to move. So skeletal muscle is what we have in our arms, our legs, in all of our limbs, which allows us to control our movement. So first we're going to look at the structure of our skeletal muscles. So each individual muscle, so we see here our whole muscle, is made up of millions of tiny muscle fibers. So you can see this is a section of our whole muscle and it's made up of loads of muscle fibers. And these muscle fibers are called myofibrils. So you can see here one of our single muscle fibers are myofibril, and here is the structure of it that we're going to look at in a minute. So our whole muscle is made up of these individual little myofibrils. And so our muscles are composed of all of these smaller units bundled into a larger one. So all of these myofibrils bundled together to form our whole muscle. However, there is a problem with the idea about this, because if we have all of these smaller units joined together, this means that there could be weak joints between all of the cells, because if we have all of these cells joined together, then all of these little links that form between all of these muscles can be quite weak if we don't have any things put in place to counteract this. And if they were weak, this means that they wouldn't be able to withstand contraction and all of the movement that we want. And so to maximize the strength of our muscles so they're able to undergo this contraction, all of these separate cells, all of these separate smaller units, these myofibrils, become fused together into muscle fibers. So this means that the joints between all of these smaller units are not weakening our muscle. We're maximizing our strength. So they all fuse together to form these muscle fibers. And so these muscle fibers share nuclei and sarcoplasm. So sarcoplasm is just the muscle cytoplasm. It's got a special name for, for any reason. It's just got a nice special name like we'll see later on in the video. So these muscle fibers, they share nuclei and they share this sarcoplasm. And this sarcoplasm is mostly found around the circumference of the fiber. And they share this so that they can fuse together. So they fuse together so they share this nuclei and this sarcoplasm to increase the strength of the joints. And so this sarcoplasm or cytoplasm of our muscle fibers has high concentrations of mitochondria and endoplasmic reticulum. And this is because it's going to need to manufacture a lot of proteins and it's going to need to manufacture a lot of ATP for the contractions that it's going to need. So that is the overall structure of our skeletal muscles. It's these large muscles branched off into these small myofibrils, which are all fused together to form our muscle fibers to strengthen the muscle as a whole. Okay, and now we're going to look at protein filaments. So we've seen that each muscle fiber is made up of these myofibrils, but what are our myofibrils made of? So our myofibrils are made up of two different protein filaments. And so one of the protein filaments that it's made up of is actin. So this is our thinner filament, and it is made of two strands wrapped around each other. As you can see here, these blue strands, they're wrapped around each other. And then these two strands are also wrapped in this protein complex that you can see here. And this protein complex is called tropomyosin, and this will have its importance shown later on. But the important thing is now understanding that this actin is the thinner of the two filaments. It consists of these two strands wrapped around each other with this tropomyosin wrapped around it. And then our second filament that our myofibrils are made of is myosin. So this is our thicker of the two filaments, and this consists of long rod-shaped tails with bulbous heads off of the side. So we have these thicker filaments with these little myosin heads poking out from our long rod. And so these are the two protein filaments that make up our myofibrils, which are our tiny smaller units that make up our whole muscle. So now we're going to be looking at sarcomeres. Okay, so sarcomeres are repeating units in myofibrils. So we know our myofibrils are these little cells that make up our muscle and our muscle fibers. And these myofibrils are made up of these repeating units called sarcomeres, and they're shown here, and we'll explain this diagram in a minute. So our myofibrils appear striped. So all of these little individual smaller units appear striated, and that is the biological term for striped. And this is because of the different bands and zones across the sarcomere, as you can see in this diagram. And we know from the previous slide that our myofibrils are made up of two different filaments, our actin and myosin filaments, and it is these filaments that cause this striated appearance. And this is because, as we can see in this diagram, our different filaments are at different areas of the sarcomere. So we have our actin filaments, these thinner filaments here, and then we have our myosin filaments, our thicker filaments with the heads poking out of them in between. And so first we're going to look at the I-band. So the I-band is the light stripe on our myofibril, and this is because it is the distance from a myosin filament to the other myosin filament. And so this means that there is only actin in this I-band. And if there is only actin, only our thinner filament, it is going to appear lighter because it has the least amount of filament there. It is the thinnest of our filament, so it's going to appear lighter, and that is what our I-band is, the light stripe. And this is because it's from myosin to myosin, so only actin is present. Our A-band is the dark stripe in our myofibril, and this is because it is the width of our myosin filament. As you can see here, it's the width of the myosin filament. And so this is where myosin and actin overlap, as you can see here. And so this appears darker because it's not only our thicker filament, our myosin, but it's also got the actin as well. So the combination of the two make it appear much darker. And that is what our A-band is, the dark stripe, the width of our myosin. And then we have the H zone, which isn't actually labelled on the diagram, but it is this little area in here. So this is a little area within the A-band, so our zone within the A-band, which is only myosin. So it's actin to actin. It's the distance between the two actin filaments where only myosin is present. And so there's no overlap. And so this is a slightly lighter region within the A-band because there is not this overlap. It's only the myosin. However, this H zone is still darker than our I-band because remember that the myosin is a thicker filament than our actin. So only myosin is still darker than only actin. So that's what our H zone is. It's this lighter region within our A-band. And then we have our Z lines. So at the centre of each I-band, we have a Z line. And the distance between two Z lines is one sarcomere. So we can see here that this area is one sarcomere. And so sarcomeres are these repeating units that make up our myofibrils. And a rule to remember which band is the light and dark is the second letter rule. So our I band is the light band because I is the second letter of light. So I band is the light band. And the A band is the dark band. And so if you remember which is the light and which is the dark, you can then work backwards and think, okay, well, our actin is our thin filament, so the I band is going to be only actin because that's how we're going to get our lightest band. And then with the A band, you think, oh, okay, our myosin is our thickest filament, so the I band is going to have to be the myosin width. And so then you can work about the H zone, which is just the lighter part of the A band. So now we've looked at the structure of our skeletal muscles, the myofibrils and the sarcomeres, we can move on to the contraction of skeletal muscles. So skeletal muscles act in these antagonistic pairs. So we have an agonist and an antagonist. And so one of these will contract while the other one relaxes. So they're able to pull in these opposite directions, causing our movement. But it's important to remember that muscles cannot push. They can only pull in one direction. It's the relaxation of it that causes the other muscle to push in the opposite direction that may look like it's pulling. But muscles can only push, they cannot pull. And so the sliding filament mechanism is how actin and myosin filaments that we saw make up our sarcomeres in our myofibrils, how these two filaments sliding over each other results in this contraction that we see here. So sliding filament mechanism is how we're going to explain how we can contract our muscles. And so first we're going to start with muscle stimulation, because we cannot cause our muscles to contract without it first being stimulated by a nervous impulse. So that's where we're going to start on how we can contract our muscles. So the first thing is an action potential comes down our motor neuron into this neuromuscular junction. So this action potential comes to the synaptic knob of our motor neuron, and it's opposite this muscle membrane. So instead of a postsynaptic neuron, we have a muscle membrane. So this action potential comes to this neuromuscular junction, and the presence of this action potential causes calcium ion channels on our presynaptic membrane to open. And so as these calcium ion channels open, calcium ions can then diffuse into our synaptic knob. And these calcium ions cause synaptic vesicles, and these are shown here, so they contain our acetylcholine neurotransmitter. They cause these synaptic vesicles to fuse with our presynaptic membrane and release this acetylcholine into this synaptic cleft. And so then our acetylcholine, our neurotransmitter, can diffuse across this synaptic cleft and bind with complementary receptor sites on our muscle membrane. And it is this binding which then causes the opening of sodium ion channels on our muscle membrane, causing sodium ions to diffuse into our muscle membrane, causing depolarization of the muscle cells, because as our sodium ions travel into our muscle cells, they lower the potential of the membrane they increase the potential of the membrane because they are bringing more positive charge into the muscle cell. And so now we're going to look about how this acetylcholine binding to our muscle membrane, causing depolarization of the muscle cells, can then lead further down to contraction. So the first thing is that our action potential that we have now triggered at our muscle membrane travels through these system of tubules. So these are extensions of the cell surface membrane. So because of this depolarization, we now have a traveling action potential, which can then travel down these tubules, these extensions. And this branches through our sarcoplasm. So this is our muscle cell cytoplasm. So it branches through down the sarcoplasm. And these tubules are in contact with the sarcoplasmic reticulum. And so this is again the muscle cell name for the endoplasmic reticulum. So it's the endoplasmic reticulum of a muscle cell is the sarcoplasmic reticulum. And this sarcoplasmic reticulum, before any of this depolarization and action potential, has previously been actively transporting calcium ions from our sarcoplasm into the sarcoplasmic reticulum, causing a very low calcium ion concentration in our sarcoplasm. And so, when an action potential travels through this system of tubules and meets our sarcoplasmic reticulum, this action potential causes the calcium ion channels to open. And because these open, the calcium ions that have been actively transported into our sarcoplasmic reticulum can now diffuse out of the sarcoplasmic reticulum down this high concentration gradient back into our sarcoplasm. And this is really important. So we've caused this depolarization of our muscle membrane, which has then traveled through our system of tubules to our sarcoplasmic reticulum, which then causes calcium ions to be released into the sarcoplasm. So now we've done with all of that, we've done with the nervous stimulation, and we have now released calcium ions into our sarcoplasm. Now we can get onto the actual sliding filament mechanism. So we can take a step back and think about our sarcomeres on our myosin filaments. They're made of myosin and actin filaments. So our myosin heads that we have here actually have a complementary binding site on these actin filaments. So you can see here. However, this protein complex that we mentioned before, this tropomyosin, is bound around the actin filament and it's blocking these binding sites. So the myosin head cannot bind to the actin filament. However, now, because of our nervous stimulation and our travelling action potential, the calcium ions that we have released into our sarcoplasm can bind to our tropomyosin, our protein complex, and it can cause this tropomyosin to pull away and free up these binding sites. So that is why this release of calcium ions is so important. It's because it is these calcium ions that cause our tropomyosin to slide up our actin filament to pull away and expose these myosin head binding sites. So now, how does this help? How does this cause the contraction of muscles? Well, if we can see in this diagram here, this is our myosin filament with our myosin head, and this is our actin filament, where we've just now moved our tropomyosin and exposed the binding sites. So these myosin heads, now the binding sites have been exposed, can form a cross-link with the binding site on the actin filament. So we can now bind our myosin head to the actin. And this bond that we form with the myosin head and the actin is weak. And because this binding is weak, the myosin head releases a molecule of ADP. And so we can see here we have a molecule of ADP, and now we have released it. And the release of this ADP causes the structural change of the myosin head. And this changes the structure so that it changes the angle. And so if our myosin head changes this angle, it pulls the myosin up the actin filament. Because it changes, it pulls on our myosin filament. It pulls it up the actin. And this is called a power stroke, the changing of the angle causing the myosin to slide up the actin filament. And this is now more strongly bound. And so, because it's more strongly bound, a molecule of ATP needs to attach to each myosin head to cause it to release from the actin. So we've done this power stroke, we've moved our myosin up the actin filament because of this angle change. We have now attached ATP, which causes the release of the myosin head. And we now have, as we said before, all of these calcium ions. And these calcium ions, yes, they free up the myosin head binding sites by moving our tropomyosin, but they can also activate ATPase, which is an enzyme which hydrolyzes our ATP. And the hydrolysis of this ATP returns it to ADP and phosphate, allowing the myosin head to change and return to its original angle. And this is really important because now it's back to its original angle. The myosin head then can continue this process of binding, releasing ADP, changing the angle and pulling up the actin filament, and then releasing and continuing. So this allows it to pull itself further up the actin filament. And so when we think of the sarcomere and our structure of the sarcomere with our filaments, what's actually happening? So the movement of our myosin as it pulls up the actin filaments actually causes the actin to move closer together, because if we're pulling it up, we're pulling the actin down the myosin as we're pulling the myosin up the actin. And so this shortens the distance between the Z lines. So we're shortening our sarcomere. So we can see here, as we pull our myosin up the actin, we're shortening the Z lines, the distance between the Z lines. And so as this shortens the sarcomeres, if it shortens all of the sarcomeres, you're shortening the entire muscle. And this is contraction. So you're pulling your myosin up the actin, shortening these Z lines, shortening our muscle, allowing for contraction. And the contraction also leads to the shortening of the I band, so you can see that here. And it leads to the shortening of the H zone, so this area where there's only myosin, because as our actin moves further in, our H zone becomes smaller. And so it's all and good that we have now contracted our muscle, but how do we extend our muscle again? How do we relax our muscle? How do we increase the distance between our Z lines again? So the nervous stimulation will stop. We'll consciously decide that we do not want to move our muscle anymore. And so as the nervous stimulation stops, the calcium ions are actively transported back into the sarcoplasmic reticulum, because there is no longer this depolarization, this action potential to open the calcium ion channels. So the calcium ions can be actively transported back into our sarcoplasmic reticulum like before. And this uses the energy from ATP hydrolysis. So remember, we had to use ATPase to hydrolyze the ATP into ADP and phosphate to return the angle of our myosin head to its original angle. And the energy released from this is then used further down the line to actively transport our calcium ions back into the sarcoplasmic reticulum. And this lack of calcium ions in the sarcoplasm causes our protein complex, our tropomyosin, to shift back and block these binding sites again. And because we're now blocking these binding sites, the myosin heads cannot bind, so contraction can no longer occur. And so when we think about evidence for our sliding filament mechanism, we think back to what we mentioned before. Because our I band shortens, this band here, this light band, our H zone shortens, which is the distance between the actin filaments, and the Z lines become closer together, so we shorten our sarcomere. And because of all of these changes, when our muscle contracts, the striated appearance, our striped appearance, changes because of the shortening of these different zones and bands. And because these stripes on our muscle fibers change, it is showing that these changes are occurring, providing evidence for our sliding filament mechanism. And one last important note is phosphocreatine. So ATP is required for releasing the myosin head, as we saw, because it's strongly bound in this new angle. And ATP is also required to actively transport calcium ions back into the sarcoplasm, our sarcoplasmic reticulum, to stop muscle contraction. But how do we get all of this ATP that we require? Because when oxygen concentration is low, we wouldn't be able to aerobically respire to produce enough ATP to do this. And phosphocreatine is this important part. It acts as a phosphate store. So phosphocreatine can quickly condense ADP and phosphate into ATP for energy. So phosphocreatine is really important. It is this phosphate store which allows us to quickly condense ADP phosphate into ATP so we can contract our muscle, and we can stop contracting our muscle by actively transporting calcium ions back into our sarcoplasmic reticulum.